Assalamualaikum warahmatullahi wabarakatuh. Today, me, Shahira, and my team members, Akira, Ashadli, and Haswan, will present on question 2 and question 4. Next slide. For question 2, which is from attachment A, is for question 2, which is from attachment 1, is regarding in silico modeling and simulation of magnetic nanoparticles for the biological cell isolation technique. Next slide. Magnetic nanoparticles are a group of nanoparticles that can be influenced by a magnetic field. In this study, the molecular modeling and simulations of the magnetic nanoparticles and the leukemia protein were done. It was done to study the isolation and removal of leukemia protein from human body that can be done through the interaction between the external magnetic field and the magnetic nanoparticle embedded molecules. Next slide. The parameter used in the molecular dynamic simulation for the leukemia proteins, firstly is the aliphatic index value. Aliphatic index value used to predict thermodynamic stability of protein. And from this study, it was found that these leukemia proteins molecules are highly aliphatic. The high value of the aliphatic index indicate that the protein molecules to be thermodynamically unstable. The next parameter is grand hydrophily value. This value used to determine hydrophilic or hydrophobic nature of the protein. From this value, it was determined that the leukemia protein was hydrophilic. Next is the half-life of the protein, in which it was found to be low for the leukemia protein. Next, the parameter for the nanoparticles. The parameter are the size of the nanoparticles, in which the increase in size also increasing the binding energy. The next parameter is potential energy surface, which is used to characterize the stable product and the intermediate. The third parameter is the enthalpy value, which is found to be negative for all the nanoparticles, indicating them as exothermic compounds. And the last parameter is the binding energy of the nanoparticles. Next slide. In conducting the molecular dynamic simulations, there are a few software that we use. The first one is PyMol. It is used to clean and purify the proteins that were collected from protein data bank before they undergo modeling and simulation in SRI's Discovery Studio software. The second software is SRI's Discovery Studio software. This software we use for the modeling and simulations of protein, where it was done with Steepest descent algorithm and CHAM phosphate. And this software also used for the study of the interactions between the proteins and nanoparticles. The last software is the SRI's Material Studio. This software used for modeling and simulations of magnetic nanoparticles. This software will optimize the geometry of the starting nanoparticles in order to provide maximum interactions and stability and to locate a minimum on the potential energy surface. In this software, the simulations and modeling of the nanoparticle were done with universal phosphate, in which the phosphate data will provide algorithm for computing the potential energy surface for a given model. The potential energy surface is important because it specifies the way in which the energy of a molecular system varies with small changes in its structure, but it's most commonly used to stimulate the dynamic of non-structure. And next, I pass the presentations to Haswan.
Okay, thank you, Sister Shahira. So, in my part, I will explain about the advantages of using MD simulations. So the first advantage is it is capable of detecting the enthalpy values of all nanoparticles. Based on the article, the enthalpy values of all nanoparticles are found to be negative. Hence, this indicates that they are all exothermic compounds. And this explains about the nanoparticle thermodynamic stability. And the second advantage is it can predict the total energy trend of all nanoparticles. For instance, when the size of the nanoparticle increases, the total energy will also increase. And the third one, it is able to calculate the binding energy for each nanoparticles, which this is very crucial in docking. So as we can see in the table 1, in comparison to the other two nanoparticles, ion 3 oxide have highest binding energy, while ion monoxide shows least binding energy. Uh, and the fourth one is it is successful in determining the best compound for targeting and isolating leukemia proteins. According to the computational studies, ion 3 oxide with a size of 1.1 nanometer is the most suitable and efficient in targeting and isolating the leukemia proteins. So that's all for the advantages of the MD simulation. I pass this to next presenter.
Okay. Thank you, Brother Azwan. So, Miss Sister Akira and Sister Aisha will be present about attachment A. In attachment A, we were required to analyze the movie and TV shows that were added to Netflix from 1993 until 2022. Okay, next. Okay, from this slide, as you guys can see, the type of entertainment was divided into two, which were movie and TV show. And there was 2 years, which were 2020 and 2021. Orange color represented TV show, while blue color represented movie. So in 2020, the number of movie that released was 517, was the number of TV shows that released in 2020 was 436. To compare, in 2021, the number of movies that was released in Netflix was 277, while the number of TV shows that released in Netflix was 315. Okay, next. So for the next question, we were required to find the number of movies released in Netflix from 1998 until 2008. So next. Next. So as you guys can see from this line graph, the number kept increasing. This shows that the higher demand of the Netflix user to watch the movie on this app. Since it is really convenient to watch the movie anytime and anywhere. Okay, next I will pass to Sister Aisha.
Alright, thank you, Sister Ahira. I am Asha Adli, and I'm going to share on the difference between the number of movies and TV shows added to Netflix from United Kingdom in 2020 and 2021. So from United Kingdom, 97 movies and 49 TV shows were added in 2020, where the differences is 48. While in 2021, 83 movies and 37 TV shows were added, in which the differences is 46. Next, the difference between the number of movies added in 2020 and 2021 is 14, while the difference between the number of TV shows added in 2020 and 2021 is 12. Lastly, the total number of movies and TV shows added in 2020 is 146, while 2021 is 126, and the difference is 26. This data will represent as the bar chart above. Okay, next. We're going to look at the number of ratings in Netflix based on the data as shown in the slide. And this data will represent in a bar chart, okay, as shown in this slide. So from this visual, we can observe that the number of ratings based on count of description, listed in, release year, title, type, and show ID are the same, which is 8807, while the number of ratings based on count of duration is 8804. Based on count of date added is 8797. Based on count of country is 7976. Based on count of cast is 7982. And lastly, the numbers of ratings based on count of director is 6173. And that brings us to the end of the presentation. So I pass the slide to the next presenter.
Okay, so for the conclusion of our assignments, we can conclude that in attachment 1, it can be concluded that the molecular dynamics is a useful method for analyzing the behaviors of atoms and molecules, which can be used in the pharmaceutical industry to determine the optimal compound for treating any disease. In attachment A task, the data analysis can be interpreted by bar graph to convey the relational information quickly as the bars display the quantity for a particular category. It helps to compare and contrast numbers, frequencies, or other measures of distinct categories of data. So that's all from us. Thank you. Assalamualaikum.